Introduction. Bacillus group include Bacillus anthracis and Bacillus cereus along with many other varieties. Bacillus anthracis causes anthrax and cereus causes food poisoning. So normally all Bacillus members are motile and not capsulated. But anthracis is non-motile and capsulated. Later you'll also study that this capsule is a virulent factor. That means it is responsible for causing infection. Morphology. Gram-positive bacilli, long rectangular rod forming chains are seen under the microscope. So here in the adjacent picture you can see long rectangular rods which were placed side to side in a chain-like pattern. So this is also called as bamboo stick appearance under the microscope. There are non-bulging spores. Unlike Bacillus cereus, there will be bulging spores there, but here it is non-bulging spores. 3 to 10 μm in length. Bacilli anthracis are the spore forming obligate aerobes. Spore forming we already learned about this. The spores formed here are non-bulging spores. Obligate aerobes means they need oxygen for their growth and survival. Non-motile and capsulated. Animal anthrax. It is most commonly seen in sheep and cattle. The mode of transmission of infection is by ingestion of spores from the soil. Clinical manifestations: fatal septicemia. That means the degree of infection is so high that it could be dangerous for the animal. Fatal septicemia. And bacilli discharged from the mouth, nose and the rectum. So this bacilli which is discharged from the infected animal often causes infection in the human. So that's why it is called a zoonotic disease. Human anthrax. The types are cutaneous, pulmonary, intestinal. Mode of transmission is by ingestion of raw, uncooked animal food which were infected by animal anthrax. Inhalation of spores, contact with infected animals. This table we are going to study about different types of anthrax. First one, cutaneous anthrax. It's also called as hide-Potter's disease because dog workers usually carry the hides and skins of the animals on their bare backs. So what happens, the spores present on the animal's skins and hide are exposed and they enter the humans through the aberrated skin. Since it's the most common type of anthrax, prognosis is self-limited. That means it is not very dangerous. That means it's not very fatal. But if it is left untreated, it can be sometimes lead to fatal conditions. Clinical manifestation. It causes malignant pustule. It is a painless black necrotic eschar surrounded by non-pitting indurated edema. Malignant pustule on the neck. It is a black necrotic eschar surrounded by non-pitting indurated edema. Unlike the name suggests, it is not malignant condition. Let's go to the pulmonary anthrax. It is also called as wool sorter's disease because people who work in the wool factory can acquire this infection by inhalation of the spores from the infected wool. Wool is taken out from the animal's body, right? Mode of transmission is by inhalation of spores. Occurrence, it is rare. Prognosis, it is fatal. Clinical manifestations, it can cause hemorrhagic pneumonia, which can lead to bacteremia, meningitis, hemorrhagic mediastinitis. Intestinal anthrax, it is very rare, and the mode of transmission is by ingestion of raw animals contaminated with spores. Occurrence, it is rare. Prognosis, highly fatal. Clinical manifestations, bloody diarrhea can be seen here. First picture is a cutaneous anthrax, the black necrotic eschar surrounded by non-pitting indurated edema. The second picture, it is of hemorrhagic pneumonia. Coming to epidemiology, it is of two types. Non-industrial cases. That means when agricultural exposure to the animals. That means when animals are exposed to agriculture, often the spores are disposed from the animal's body into the food. That's how you can acquire the infection. And industrial cases, it is by infected animal products like hair, hides, bristles and wool. Pathogenesis, it is due to the virulent factors. So in Bacillus anthracis, there are two virulent factors. First one is its toxin and second one is its capsule. Coming to the toxin, it is controlled by a plasmid PXO1. So under toxin, you have to remember three factors. First one is protective factor, edema factor, lethal factor. So as the name suggests, edema factor can lead to the edema. Lethal factor can cause cell death. Now the protective factor is the one which binds to the receptor and allows the edema factor and the lethal factor to enter the cell. So this is about the factors. And the toxin is controlled by plasmid PXO1. Coming to the capsule, it is controlled by PXO2 plasmid. This capsule of Bacillus anthracis is particularly made up of polyglutamate polypeptide instead of polysaccharide. So the function of the capsule is to inhibit complement mediated phagocytosis to survive in the human body. So to simply summarize this pathogenesis, it is due to the virulence factors like toxin and the capsule. Toxin is regulated and controlled by PXO1. Capsule is controlled by PXO2. So under toxin, you have to write three factors: protective factor, edema factor, and the lethal factor. Protective allows the entry of other two. Edema causes edema. Lethal causes cell death. And under the capsule, it is made up of polyglutamate polypeptide, and it inhibits the complement mediated phagocytosis. So this is about pathogenesis. Lab diagnosis. One way of doing this is by specimen collection. Pus is collected in case of malignant pustule. CSF is collected in case of hemorrhagic meningitis. Sputum in case of pulmonary anthrax. Direct demonstration is another way under which gram staining can be done. So long rectangular rods forming chains are seen under the microscope. In Mcfadden's reaction, Bacillus anthracis is stained with polychrome methylene blue for 30 seconds. Amorphous purple material surrounded by blue bacilli is observed. That means, remember this way: Bacillus anthracis, when it is stained with blue stain, it becomes blue, right? So remember this word, blue bacilli. Now it surrounds the amorphous purple material. This is the Mcfadden's reaction and it is important. Coming to another method, which is by direct immunofluorescence. So on the capsule of Bacillus anthracis, there will be antigens present. So antigens, when exposed to antibodies, they attach to each other. Instead of using normal antibodies, we use fluorescent type monoclonal antibodies. So when these get attached to the antigens, we can see them under direct IF method. Another method is ascoli thermoprecipitation test. Ring precipitate is formed when specimen is in putrid form. Let us see the pictures for all of this. The first one is Mcfadden's reaction. Purple color amorphous substance is surrounded by blue bacilli. Second picture here is the ascoli thermoprecipitation method. There will be a ring precipitate formed in middle. Antigens and antibodies are fused together in the zone of equivalence. There will be a visible precipitation. Third picture is a direct IF test. The fourth one is a bamboo stick appearance case of gram staining. Now coming to the culture smear, it can be done by gram staining, bamboo stick appearance we just saw. Pore staining is demonstrated by Ashby's method. The special stain used here is malachite-free stain. Molecular diagnosis is done by under cultural characteristics, first we'll study about different types of agar on which this Bacillus anthracis can be grown. It is nutrient agar, blood agar, gelatin swab agar. Under nutrient agar, when we view it under the low power microscope, we can see the medusa head appearance. So under blood agar, we can see the dry, wrinkled, non-hemolytic colonies of Bacillus anthracis. Under gelatin swab agar, inverted fir tree appearance will be there. In this pictures, this test tube picture is a gelatin swab agar, inverted fir tree is seen. See, this is the fir tree. Inverted fir tree appearance will be there in gelatin swab agar. In case of blood agar, there will be non-hemolytic colony formation of Bacillus anthracis. Under this picture, we can also see the medusa head appearance of Bacillus anthracis in case of nutrient agar. This is about different types of agars. Coming to selective media. Solid media with penicillin gives string of pearl appearance. Blood media is also used, which is made up of polymyxin, lysozyme, EDTA, thallus acetate. Remember all of these cultural characters. I have a mnemonic. Medusa wearing pearl-plated necklace sitting under fir tree with blood on her wrinkled neck. Medusa here refers to the medusa head appearance under nutrient agar. Wearing pearl-plated necklace. Pearl refers to solid media with penicillin, and plated refers to plate media. Right? So this both come under selective media. So Medusa wearing pearl-plated necklace sitting under fir tree. Fur refers to inverted fur appearance in case of gelatin swab agar, with blood on her wrinkled neck. Blood on her wrinkled, this both words, blood and wrinkle, refers to blood agar, in which dry, wrinkled, non-hemolytic colonies of Bacillus anthracis are seen. So this is a mnemonic for cultural characteristics. The first picture here is a pearl appearance in case of solid media with penicillin. The second picture is a red agar, non-hemolytic colonies are seen. The third pink color in which you can see is a medusa head appearance. The fourth test tube picture is a inverted fir tree appearance in case of gelatin. Now coming to the treatment, for the cutaneous anthrax, we give an amoxicillin, deoxycycline for 10 days. For pulmonary or the intestinal anthrax, antibiotic regimen is followed along with the anthrax immunoglobulin. Vaccines are available, that is called Biothrax. Post-exposure prophylaxis, deoxycycline for 60 days along with the vaccine is given.